Hello. Hello from Baltimore, from Johns Hopkins University, where I have to give my talk, unfortunately, remotely. I would have loved to spend my time with you in Italy, where I typically spent my summers since working there from 2002 to 2009 for the European Commission. COVID-19, is it a brain disease? This was one of the questions we posed. And at this time, in February, March, it was actually becoming obvious that more and more patients were showing neurological symptoms. So we tried to understand, is there possibly a contribution of brain infection? And this is easier said than done, and I will tell you a little bit about our story. But before doing so, I think we should state very clearly, I'm representing a Center for Alternatives to Animal Testing, but I'm a physician working in the School of Public Health. And this protective gear saying saving more than animals, I think, is getting the message across. We want to help patients and consumers when moving away from animal testing and using advanced methodologies. This was, by the way, the title of an article in 2005 of the Scientific American, which my predecessor, Alan Goldberg, and myself did write about our area of work. But to get started, COVID-19 is so far an incredible success story. If you think that the first cases were actually reported in end of December of 2019, on the 31st the WHO was informed, but already on the 10th of January the first PCR was available out of the UK. On the 11th of January the whole genome was sequenced, and on the 17th of January the PCR, which later became the WHO method, was available. In March already several antibody tests became available, and on 1st of May the first FDA licensed emergency use authorization for a drug which had shown clinical benefit was there. If we for a second compare this to HIV, where it took us years to identify the virus, where it took us many years, decades, to come to meaningful treatments, and otherwise I think this is really a remarkable success story which we all have witnessed over the last 9 months. Let's talk for a moment about the diagnostics of COVID-19. We have essentially two methodologies available, PCR and antibody serologic methodologies. And if you looked around April into the quality of these methodologies, their sensitivity, so how many of the positive cases they miss, and their specificity, how many of false positives they're generating, this all was around 93%. So we can say the balanced accuracy, or BAC, of these methodologies is 93%. What does this mean when I'm actually now looking into a patient? In the US at the moment, we do have about 40 cases in 10,000 people, 40 in 10,000. That's what we would pick up with our assays. If the assay is 93% accurate, this would mean that of 10,000 tested people, we will identify 37 of these 40. But on the expense of 7% false positive, which is 700. So the probability of a correct result is actually only about 5%. And we should be very clear, these assays at the quality which we had around April were obviously problematic when applied to larger populations. The negative results, however, we could largely rely on 99.9% reliability of these assays when doing the exact same calculation. So one of my big worries is that actually we might have a situation where a lot of these false positives were actually the asymptomatic carriers we were identifying early on. And it could very well be that these strange findings of no antibodies after diagnosed COVID-19 infection, reinfection after the first one, could actually be prompted by false positives. And we don't know how many people believe they are now immune superheroes which cannot be infected again, who actually were false positively diagnosed. So this is a real concern applying these type of methodologies. But I would like to move to another area of this, which is the medical challenge of developing drugs and vaccines. And this as fast as possible, please. So the normal development times of about 12 years for a drug, 10.5 years for a vaccine, they should be shortened now to months or, if at all, few years. And the response of the medical community was incredible. Using a lot of these fast-track methodologies, a lot of pragmatic shortcuts, we have seen that by now we already have more than 10,000 clinical studies launched. 2,600 and more are actually interventional studies. That's what the Cochrane register shows. And we have already more than 160 vaccine candidates, some of them in actually advanced development. In April, there were already 115, so just 4 months after the whole disease started. That's an incredible contribution of science which took place here. And part of this is possible because we are not using the traditional animal models. We are still struggling in identifying proper animal models for this disease, but we are using a lot of new approach methods, both towards enhanced clinical trials, but also in cell culture and computational methodologies. This article I contributed to is available as open access, and it discusses drug repurposing, target discovery, drug efficacy testing, and others, and what these new type of methodologies can do for us. And one thing we should be very clear about, cell culture is not what it used to be. When talking about cell culture, most people think of the petri dishes with cells in a monolayer. They look like pan-fried eggs in a pan sunny side up, hardly having any cell contact, very little cell density, only about 0.1% of a real tissue. But what the last decade has given us is organotypic type of cell cultures, the organoids, the organ-on-chip systems, the microphysiological systems. And these developments really give us for the first time human tissue equivalents which are healthy, don't come from the surgical material of a diseased patient, and they can actually reflect the architecture and the functionality of some of these organs. We held 2 stakeholder workshops, they are given here, published in 2016 and 2020, which summarize the state of the art. Again, they are available online in our open access journal Altex, and I refer you to these to learn more about this. And the expectations are enormous. To give you only one testimony, this is Dr. Francis Collins heading the NIH. He said in 2011 already, with earlier and more rigorous target validation in human tissues, it may be justifiable to skip the animal model assessment of efficacy altogether. So drug development without animals. An early caller. In 2016, when testifying before Congress, or Senate actually, he said, showing one of these organ-on-chip devices, I predict that 10 years from now, safety testing for newly developed drugs, as well as assessment of the potential toxicity of numerous environmental exposures, will be largely carried out using human biochips, and will mostly replace animal testing for drug toxicity in environmental setting, giving results that are more accurate, at lower cost, and with higher throughput. And you could have testimony from heads of FDA or EPA and many others, and you might have heard that EPA decided last September to move out of animal testing by 2035. So the expectations are enormous. In this context, I would like to introduce to you our mini-brain project. Our mini-brain project started with funding from FDA and then NIH NCATS. And in 2016 at AAAS, we were able to announce for the first time that we were able to mass produce small balls of cells. You see these balls of cells here. They're about 350 micrometer, 30,000 cells. And it takes us about 3 months to form thousands of these. One cell culture plate gives us about 500 to 800 of these balls of cells, which contain astrocytes, oligodendrocytes, and essentially all types of neurons we ever looked for. And they're communicating spontaneously. And the important thing is really that these are identical from one mini brain to the next, from one week to the other. And this is a tool which lended itself now to ask the question, are symptoms of neurological manifestations of COVID-19, which were seen in 30 to 60% of the patients, depending on the study, are these type of symptoms perhaps also caused by direct infection of brain cells? There's enough explanation by the severe inflammatory reactions, the possible coagulation of blood vessels and others to explain these, but it could very well be that there's also a direct infection. Our mini brain model had earlier served us for some virus infections. In 2018, we published on dengue and Zika virus. We also have work on HIV and JC virus, because these models can help with a human pathogen to provide the human tissue in order to demonstrate infectability. So our own research was now addressing is SARS-2 COVID among the neurotropic infections. And this was prompted by these findings that already in February, reports as China said 36% of the patients have neurological symptoms. And there were even case reports of fatal infections of the brain, encephalitis, by the virus directly. And we knew from the literature that other coronaviruses can infect brain cells. So we teamed up with Bill Bishai and Corinne Bullen from infectious diseases here in Hopkins, and indeed we were able to infect the first brain cells. This was the first human brain cell which I have ever seen. My wife, Lena Smirnova, who is also the senior author of this work, she took this picture, this is the original, taking with her iPhone, and sent me from the confocal microscope home this picture. See, this is a brain cell full of viruses. And these are hundreds of viruses, because each virus which infects a cell replicates more than 100 to 1,000 times and then lyses the cell and starts infecting others. This was covered very early on by the Financial Times, you see here, and about 140 other outlets followed, because this has really news character, because it changes to some extent our view on this disease. This is not a lung infection which affects the bloodstream downwards, but it can infect one of our most precious organs directly. We published this shortly after the peer-reviewed publication. This is our team, my own team to the left, and on the right, Andy Pekos, one of the SARS experts in the world, who is just one floor above us here in Hopkins and helped us a lot. And you see here my own team, they have good reason to look into these type of systems. They're interested in DNT and DIT, which stands for developmental neurotoxicity and developmental immunotoxicity, because our big concern is always, is the embryo affected? And this is one of the big questions we are having at the moment. What you can see here from their work is virus-infected cells in red and in green, demonstrating with different technologies that the virus is found in these human brain tissues which we are generating after infection with very low doses of virus. What we also could show is that the virus multiplies here by a polymerase chain reaction, PCR, so more than 100-fold increase in 3 days of infection, which is in line with the virus cycles. In the beginning, we had some problems that also we still, after washing our cells, we found tremendous amounts of virus in the supernatant. So this was not very satisfying. So we improved our experiments and we moved the mini brains actually into such Eppendorf vials, and we were able to wash them much more rigorously. And now you can see the repeat experiment where again we saw a tremendous increase of virus both to the left in the virus-infected mini brains and in the supernatant. So these cells are lysed and the virus is freed and can possibly infect others. So the virus seems to hit the brain. We also were able to show that the angiotensin-converting enzyme receptor ACE2, which is the receptor the virus needs for entry, is actually present in these human brain cells. Quite worrying, they're also available in the early stages, the neuronal precursor cells. So these forms of cells of the developing brain are also infectable with virus. And two technologies showed us clearly that the virus is replicating within these cells. This is only a start, a first step, because as you can see here, only a few cells in red to the right are virus infected out of the about 30,000 cells which we are having in one of these brain organoids. You can only see one layer here, so this is not a reflection of the entire brain, but still it is a very small proportion of cells which is affected here. And it seems that more cells are being infected as the infection pursues. At this moment we are looking into many things. What are the cells which are infectable? Why are these and not others infected? What is the pathology which is being induced by this? Is the blood-brain barrier possible protecting many of us, though in the acute virus infection, strong inflammation, a cytokine storm, this is likely to break down. But most importantly for us, is there indication for disrupted neurodevelopment? As others have shown, the virus can pass the placenta barrier, and this might have impact on the developing brain. Another consequence of this could possibly be, if the brain can be infected, are we ever getting rid of it again? We know that virus infections fuel, for example, the probability of developing Alzheimer. Could they possibly contribute to neurodegeneration? But the brain is an immunoprivileged organ where immune cells spare these cells typically, and we don't get rid of virus infections ever. So you see from these discussions, this is one humble contribution to an enormous wave of scientific publications. On the 19th of August, when I made this presentation, I found almost 42,000 accepted scientific publications in PubMed. Add to this all the self-published articles and archives. It is enormous how the scientific community has responded to this and has flooded us with information. This will obviously give us some problem that we need to sort this material out. We will need to systematically review this. We will have to use approaches of quality assurance and scoring and condensing this information. And I have a chair for evidence-based toxicology, but I think what we are needing is now an evidence-based virology as well. So, coming to an end, I think what we are witnessing at the moment is a scientific gold rush at Klondike. Anybody who can spell COVID-19 writes a grant or a paper. Most of these people start the endeavor ill-equipped because they don't know these viruses, and few of them really are successful and get rich. Few of them actually get rich. These are typically those who sell the shuffles and the sieves, those which are selling us the reagents. But I think still we are finding a lot of gold, and I'm impressed by the scientific response to this, and I'm impressed by the scientific response to this pandemic. So, coming to an end, I hope I could show you that alternative methods are enabling technologies. The development, the building of capacities on alternative methodologies, brain cultures in our case, but in similar ways lung cultures, gastrointestinal organoids and others have been used to actually come closer how the virus is impacting on the pathophysiology. So I think our investment in alternative approaches helps to solve COVID-19, and I hope that we take advantage of these changes and use them also as a door opener for broader using these methodologies and overcoming some of the shortcomings of animal experimentation.